Well, good afternoon, everyone, and here we are on to yet another exciting session at LitFestx. This time with a guest from overseas. Please welcome Mr. Jon Turney, who's live with us from Bristol in the United Kingdom. Whilst John's book is a bestseller and is famous, I would also take this opportunity to introduce him. Jon Turney is a science writer and a former features editor of the Times Higher Education Supplement. He has taught at Imperial College and was head of the Department of Science and Technology Studies at UCL. He was also a popular science editor at Penguin Press and is the author, among much else, of The Rough Guide to the Future and The Lovelock and Gaia. He's also the editor of A Quark for Mr. Mark, which is 101 poems about science. John's latest book on superorganisms is a very interesting piece of work, and I'm going to dive straight into this very interesting book and topic, John. What does it mean to be a superorganism? And you've devoted an entire book on first describing what is a superorganism and what qualifies one to be a superorganism. So how does one arrive at this word, and what is it all about?
Okay, well, superorganism, I suppose, is my solution to the question of what do we call a creature that isn't an assembly just of its own cells. And a human being has so many trillions of cells, as people probably know, but along with those there are lots and lots of others which live in and on us. Some bacteria, lots of viruses which we know less about, some fungi, and it turns out as we study them more closely, as we have done the last couple of decades, that they are very intimately involved with a great many of our physiological, biochemical and other functions, so that the whole system in a way functions as a whole, that we're certain that they're not just sort of living on us casually as organisms who graze on human byproducts, but they supply us with some useful things. We interact with them very closely all the time. They affect us in lots of ways that we're beginning to understand more clearly. What do you call that? I'm a person. I have a body, but I'm also, it turns out, a sort of shambling mass of bacterial ecosystems. Various other terms or metaphors have been thrown into the mix, so that I could think of myself as a giant ecosystem or a collection of ecosystems. I could be a kind of a bioreactor, like a laboratory flask full of broth, but actually in my stomach. Or I could be a holobiont, it's been suggested recently, which is a rather more scientific sounding name, but I decided that actually superorganism is the easiest one to get your tongue around, and is the most meaningful way to think of it. It doesn't mean super in the sense of super special, because as soon as you start thinking about this, you realize that bacteria are everywhere. They're always and everywhere there first. So all the other larger multicellular creatures have grown up in and around bacteria as well. So essentially everything which is larger than a small bug, is a superorganism also. So we're one among many.
So we're a superorganism amongst many other superorganisms.
Yes, all of the creatures big enough to see, which are multicellular, have some microbial load. Ours is probably larger and more complex for reasons that we might want to go into. But to say that bacteria were around for several billion years on the planet before anything else, so everything has to have found ways of coexisting with them.
So so why does it then make sense to think of humans as a superorganism? I mean, since you mentioned that there are several others, what aspect about human beings and the human body would you specifically state towards us being qualified for a superorganism?
Well, I suppose the qualification, as you put it, is the number, the diversity, and the importance of the bacteria and the other microbes. Numbers first. I mean, you often read short articles that say there are 10 times as many bacterial cells in a human as there are human cells, and they're both number in the trillions. That's roughly right. We could argue about the precise number, but they're certainly the same order, and there are probably more bacteria. Of course, they're very small, so they don't outweigh us in mass, although the bacteria in your and in my large intestine probably weigh a couple of kilos. So it's a pretty big bowl of broth if you put them all together. But more important than that, there are many, many different species of bacteria. So although each individual small microbe only has a relatively small piece of DNA and not so many genes, it turns out that they have many, many genes. Bacteria have been out there in the world evolving very fast for billions of years, and they've explored, shall we say, the possibilities of metabolism, of chemical transformation, in a much more extensive way than we have, and we take advantage of that. People probably remember that the Human Genome Project worked out all of the DNA in human beings, in each human cell, and in sequence about 15 years ago, and we know how many genes there are in each human cell. Each cell has the same complement of genes, and there are maybe 20-something thousand of them. 23,000 is the figure that's now normally quoted. The bacteria in your gut have 2 million genes, so it's a couple of orders of magnitude more. And they're genes which allow the bacteria to do things which we can't do or choose not to do. One very well studied example, I mean, it's difficult to give a summary of the whole complex picture, so one tends to look at individual bacteria, which then you pull out thousands of them and study them one at a time. There's one called, let me say this slowly, Bacteroides beta theta iota omicron, which isn't a very useful name, but it has something like, it lives in the gut, in the large intestine, nowhere else as far as I know, and it has something like 260 different genes for enzymes which help break down complex carbohydrates, the fiber that you might eat with your, as we call it, you might eat with your meal. Human cells don't bother to make nearly so many. They make a few tens of enzymes of that sort. So B theta, as I call it for short, is kind of seriously specialized in digesting things which we eat and helps us to derive energy and other things from them. And it turns out, if you study it more closely, that we encourage it to colonize our gut. The epithelial cells in the large intestine secrete a particular carbohydrate which has a tasty sugar residue on the end, which this bacterium can nip off and get a quick energy hit. So we sort of provide it with offer it a tasty morsel to encourage it to come along and live in the cozy confines of the gut and help us digest the rest of our food. And there are other interactions between us and that particular one, which I could go into, but it's a very intimate and useful companion. And there are hundreds, thousands of bacteria, which we haven't studied in such detail, but most of them seem to do something quite useful. And
you spoke a little bit about the usefulness of different kind of bacteria, and we know that they help us with our digestion and sort of build up our immunity, so on and so forth. But how has the human microbiome changed? I mean, what has been the evolution over the years?
Excuse me. Hard to know because we can't go back and look at what the microbiome used to be like when humans first came off the savannah or wherever it was that we evolved. The way that there are a couple of ways of studying the microbiome now and in the past. Now, typically, people look at mice. You can rear mice without a microbiome. Essentially, you deliver them in a sterile chamber by cesarean section, and then raise them very carefully, germ-free, and then you can put microbes back and see what effects that has. If you want to look at the whole microbiome, you tend to be getting into rather messy areas, looking at samples of feces, and we can look at many samples from many people. One of the areas that people have explored is looking at the microbiome of other primates, which might have a diet which resembles more closely what our evolutionary forebears had to deal with. And also, of course, you can look at modern hunter-gatherers who are somewhat isolated in some cases from the diets that we like to call civilized, and certainly tend to have very different eating habits. And the suggestion is, I don't think it's firm yet, that on the whole they have, certainly people who eat a great variety of plants, for example, and less meat than I would as a typical Westerner, have a more diverse microbiome than someone who lives in a British city like I do. The actual mix of species is difficult to pin down because it's so diverse between individuals in all communities. There isn't, it looks like one typical or obviously healthy or balanced, as the term that often comes up. We don't quite know what balance means in this context. There isn't a typical healthy microbiome. But it looks as though dietary change does make a change. Whether that's good or bad is also not clear-cut. There are also other things that we've done much more recently, apart from inventing agriculture and starting to rear cattle. The advent of antibiotics obviously looms large in this context because we've waged war on bacteria, thinking that they're bad and they're germs and they're our enemies for the last century or so. And we wouldn't want to be without that. But when the average infant in the developed countries has half a dozen or maybe 10 doses of antibiotics before they go to school, then the development of their microbiome must be affected, surely. And even more strangely, in terms of the rest of human history, we quite often now deliver babies by making an incision in the wall of the uterus and removing the baby by hand rather than going through a normal labor. And it's clear that although the baby in utero is probably sterile, more or less, a lot of microbes are acquired during that normal delivery. So babies who are born by cesarean section end up with a microbiome that looks like any other child by the time they're 3 or 4, but they probably get there by a different route, and at the very early stages, their microbiome may well be rather different.
Right. And in terms of all the research on the change in microbiomes in the human body, where is that leading?
I guess, like most biology, most biomedical research, we find out these things in order to do something, to intervene. There are some medical conditions where it's clear that we can already intervene in the microbiome. We tend to do it rather crudely. There's a procedure known as a total fecal transplant, where if you're an elderly person who has a very bad infection with the harmful bacterium called Clostridium difficile, treating it with antibiotics tends to be ineffective because you wipe out the rest of the microbiome, and you may kill C. difficile, but it comes back more quickly than the others. And quite a lot of cases of that really very severe condition have been treated by actually giving people a transplant of feces, which is to say, transfer the entire ecosystem from another person in a way that doesn't cause any contamination anywhere else, transfer it into the bowel. That does some good. If we knew a little better what we were doing, we would transplant not the entire ecosystem, but some suitable selection from it, and that might do some good for people with a range of bowel conditions and possibly some other medical problems. Then there are a lot of people who think that you can assist the cultivation of your microbial garden, if you like, by your diet, by eating prebiotics, which are foods which bacteria particularly like to eat as well as us. So often complex carbohydrates, in particular kinds of plant food would be indicated there in your diet, which most of the planet still eats in any case. It's a little disappointing when you go into the detail, because the dietary recommendations tend to look pretty much like the ones that we hear anyway for healthy eating. Avoid processed foods, don't eat too much red meat, eat lots of plants, not too much of anything. And then there's a quite interesting mid-range between the total transplant and just eating different food, which is eating food which has bacteria in it, what we call probiotics. So that would be the fermented foods, which tend to vary between cultures, but most cultures have fermented products that they like to spice up their diet with. I eat yogurt for breakfast every day. It's full of bacteria. I don't know. I like to think it's doing me good. The clear indications yet aren't that sure, but it certainly seems to do no harm. Whether any of those bacteria actually get down through the acid in my stomach and end up as part of the colonies in my bowel, I haven't actually checked. But when we know more, there will probably be probiotic preparations, maybe some that are good for everyone. There may well be some that are tailored for particular conditions. The far end of speculation here is that some bacteria actually are involved in chemical interactions in the body which affect brain states, for example, affect mood, perhaps even anxiety states or depression. So it's not impossible that one day someone would take a particular probiotic at breakfast time to help lift their mood. But that is some way off, I'd say decades, not years.
Very interesting, Jon. From a lot of this new sort of wave of research coming in, for you, what is that one sort of big insight, that one big new scientifically important result that has come out?
So many to choose from. I mean, the field has really exploded in these last 15 years. Once we learned how to sequence the human genome, we got some fancy technology out of it, which allowed us to sequence masses of DNA absolutely everywhere. So lots of people doing this, vast amounts of data being processed now more slowly. For me, I think it's a conceptual thing. I've always been very interested in the immune system and how this apparently very complicated apparatus, which defends us against, certainly against the bacteria we don't like, against pathogens, against invaders, as we call them, against infectious disease. Why it is extraordinarily complicated whenever you read what we know about how it works. Why is it so complicated? And there's a suggestion, which I think seems intuitively very appealing, that one of the reasons for that is that we and a number of other higher vertebrates, if we think of ourselves as higher, maintain a very complex microbiome. So our immune system has to be particularly nuanced and sensitive. It has to act with a lot more discretion than something which is just a simple defense system that says, you're a foreign organism, you're bad, get out, we must eliminate you. There are diplomatic relations that have to be maintained with the microbiome in the gut. And it looks as though the complex parts of our immune system didn't evolve in vertebrates until they began to acquire microbiomes this complicated. Other creatures, as I've said, we could still think of as superorganisms, but until you get to the vertebrates, they tend to have much simpler microbiomes, also much simpler immune systems, which are more simply a unified defense and surveillance mechanism. So there's an interesting evolutionary tie-up there, I think, and it's been a very complicated problem to understand, so it simplified it a bit for me.
very interestingly, you mentioned about the immune system, and some of the new research which can lead us to better manage our immunity and hence our overall health. Um, in terms of the applications, uh, I'm also talking about, as you mentioned, the number of antibiotics that today even children have to take or end up taking rather. Do you think broadly this can help us get out of this circle of pumping our bodies with antibiotics at the slightest chance of a fever or a cold or whatever it is, and broadly also, what other applications do you think this research can lead to?
The antibiotic question is tremendously difficult. I mean, they are miraculous. Millions of people no longer fall down dead from diseases which once would have been the end of their lives, and the drugs work. They inevitably pitch us into an evolutionary arms race with bacteria, because if you use a broad spectrum antibiotic, you're exerting an immensely strong selection pressure on something that reproduces very fast, so they develop resistance, and that story began more or less on day one of antibiotic use. So quite apart from the effect on our personal microbiome, there's every good reason to cut down on antibiotic use as much as possible. As I said, no one would wish to be without them. There may be more occasions when you want to rein in the prescription of antibiotics and say, well, let's just see if this clears up. Maybe your child has a small ear infection and it's distressing for them, but if you think of their long-term prospects, maybe this time we won't give them a dose. And of course, in many countries, we do this completely mad thing, which is feed antibiotics to livestock to allow them to grow faster. So the world becomes a dilute solution of antibiotics, which is completely mad, and the farming industry should just stop doing it. But getting that through to political lobbying and regulatory system is a long and tedious argument, which is still going on. And yes, people, I think, will, if they learn to love their microbiome as they might, they will perhaps not reach for antibiotics so regularly, go to the doctor, and if they're feeling a bit under the weather or they have a cold, which of course is a viral infection, so antibiotics won't have any effect anyway, not press the doctor to send them away with a week's dose of antibiotic. And other applications, as I say, I think will come later, and they will be more in the way of cultivating particular bacteria rather than avoiding broad spectrum damage. But we haven't yet done it's easy to dig out the genes, and you don't even necessarily know which bacteria they come from. You then have to study the individual bacteria, you have to cultivate them, you have to do controlled studies on administering them to people and seeing what effect they have. This all takes a lot of time, takes a lot longer than putting a sample into a DNA sequencer. So that's really more specialized, more laborious, more meticulous work. And we will see treatments and preparations coming through. There are already suggestions of antibiotics you might want, bacteria you might want in your mouth to help prevent tooth decay by outcompeting the bacteria which live on the tooth enamel and make holes in it. Similarly for skin preparations, maybe anti-dandruff preparations which help keep down the bacteria which produce dandruff from the skin by providing other bacteria that colonize those niches. So those sort of superficial, as it were, surface treatments, I think might happen first before we deal with the internal ones so extensively.
Very interesting, John. So essentially what you're saying is we could see a time where instead of having antibiotics fight, let's call them bad bacteria, you will have custom-made good bacteria for specific applications of the parts of the body fighting the bad ones, or let's say, overtaking them.
Or helping to keep them away. Yes, when I mean, I don't think there's any prospect of having, if you have a serious bacterial infection, and one must think of this in terms of compartments. I've said that we contain all these trillions of bacteria. They're in a sense still kept outside because I am as you are, basically a tube. So the gut has an inside and an outside, and the bacteria are on the outside. They're inside the gut, but they're outside the body tissues. If you get an infection inside the body tissues, then you're probably going to want antibiotics to make it go away as fast as possible. But there may well be bacterial preparations which are used for other things. And the idea that we're not trying to eliminate them, but to tune the combination that colonize. So that, if you take all the bacteria out of the gut, all you're doing is leaving it open to colonization by the next thing that comes along. And bacteria reproduce very, very quickly. Maybe every 20 minutes they can double in number. So the wrong one can have a very rapid population explosion under the right circumstances. That's the kind of thing we're trying to prevent by keeping. As I said, I'm not sure what balance means, but I will use the word balance in this instance by keeping a balance of populations in the gut, which is where one tends to focus on, because although we have sort of mini microbiomes in many places, the vast majority of our bacteria are in the gut and in the large intestine.
Wow. Some really interesting insights from you, Jon, and all the very best for your book. I'm sure it will become a good piece of work for people across the world, not just in India and UK, to understand our inner system, and also to understand what is the kind of developments, research, and hopefully new insights that could help us lead a better life. And like you mentioned, maybe
we can only hope.
and like you mentioned, maybe to begin by watching our diet and our breakfasts and lunches better.
Sure, let's all do that at the very least.
So thank you so much for being on LitFestx, Jon, and I'm sure our live viewers, as many more who will watch this video going forward, will have a lot to learn, and will hopefully refer to your book for further information.
Thanks very much. Great to talk to you.
Likewise. Bye bye.